We take it for granted that a single cell will seamlessly divide, multiply, and assemble into complex, functioning tissues. But sometimes that progression stops. The march toward adulthood simply halts, freezing the organism at an immature stage indefinitely. This isn't death. The cells continue to consume energy and perform basic functions, but they are permanently barred from maturation. This suspension shows that growth is not a passive, automatic result of being alive. It is a gated process that requires an active command to continue. This microscopic image shows C. elegans, a translucent worm that serves as a primary model for scientists studying these developmental pauses. Researchers observed that this worm grows through distinct larval stages, each driven by highly precise sequential bursts of gene expression. While science identified these genetic pulses years ago, the physical timekeeper coordinating their rhythm remained a mystery. We lacked the identity of the central conductor. Without a mechanism to dictate the exact start and stop times of these pulses, the transition to the next stage of life is impossible. Using DNA sequencing films like this one, alongside protein analysis and AI modeling, researchers at Cold Spring Harbor Laboratory finally located that timing mechanism. They identified a feedback circuit formed by two specific proteins, MYRF-1 and LIN-42, which together act as the master developmental clock. Most biological cycles, like the circadian rhythms that govern sleep, operate in continuous, repeating loops. This circuit functions instead as a molecular ratchet. It allows a genetic pulse to occur, then physically locks behind it, ensuring development only moves in one direction. This non-repeating mechanism is vital. It prevents an organism from accidentally repeating a growth stage or regressing to an earlier biological state. MYRF-1 initiates each developmental stage, prompting an intense burst of genetic activity. It then activates LIN-42, which regulates the pulse's intensity and duration. At the end of the sequence, MYRF-1 acts as the final checkpoint, resetting the system for the next step. When researchers intentionally blocked the MYRF-1 protein, this entire sequence collapsed, development hit a wall, unable to progress to the next phase. MYRF-1 is the mandatory signal for growth. Without it, the biological doorway to each subsequent life stage remains permanently closed. The mechanics of this timing circuit in a microscopic worm offer a direct window into how human cells differentiate and mature. Failures in this master timing system provide a new model for understanding the structural causes behind severe human developmental disorders. In the future, this research may allow us to address genetic diseases by specifically targeting and repairing these internal cellular clocks. Decoding this microscopic timekeeper provides a roadmap for identifying exactly where and why human biological progression derails. This timing circuit operates independently within every single cell of the organism, rather than relying on a central command center. Across millions of cells, these independent biological clocks maintain perfect, simultaneous synchronization without ever experiencing a single detectable failure. Scientists have yet to determine if these cellular clocks are actively communicating with each other or if they simply share an incredibly precise internal rhythm. Human life depends on this silent, synchronized ticking, happening inside every cell, perpetually waiting for the signal to move forward.